Okay, guys. Physiology. We're going to start right on time, and I'm going to end at 7 until 2 so you can pee-pee. Okay, so this one is, it smells like Doritos in here. Are y'all just already smashing? Okay, you had an exam yesterday. Has the weariness set in, or are you still pretty excited? You're excited or what? Are you watering yourselves? And not with just beer, not only beer. And feeding yourselves, kind of thinking protein. Really do remember to put your body first, and this all will come much more easily. Truly, truly it will. I'm using one of these. I'm going to use it. There it is. This is, I don't know, Genom's medical physiology, and it's in your little bill, your review accesses and things in all the books. This is what I'm going to for all of physiology. I'm using it in order so that you have that kind of reference. I don't love it. It's very, I feel like it's translated from another language, so it's like missing like some prepositions, you know, that would make it make sense. But it's all there, and you can do what you do, which is Google other stuff. But I am using, I'm following it. There is a chapter summary at the beginning of each of those, and if you're just that person that hates to read text, but you like some bullet points, those reviews are really good. Okay, so the first thing we're doing in this section is the visual systems, next is auditory, and number 9 there means chapter 9. Okay, so I'm a biology nerd, and I love the little details, and you'll see that here in a minute. But I love the whole big picture and how it all fits into that circle of life. And we see because there's a sun, which is just cool. And you know, every single thing you ever learned in biology, your whole body is just doing all that simple stuff. And I just think that's so cool. And it's so impossible for me to believe, no matter how much you know you study this stuff, that that works. That there's like solar light, and then there are like plants, and then you can see. Okay, but hold on to loving that because it gets worse, right? So anyway, there's the and if you think about it, and if you've studied, you know, creation, I believe in all kinds of things, but if you look at sort of a linear thing there in Genesis, it's interesting because light really kind of came first, and then the other things were adapted because of light and water. And whether you buy or don't buy or whatever doesn't matter, but it is interesting that really geologically makes sense. And so plants are producers on the planet, and they fix the stuff that's in the atmosphere that's free, solar energy and carbon dioxide, and they make their own glucose. So we do something real similar in terms of using the solar energy, but we're not making glucose with it, although that'd be kind of, but we are using it then just to be able to see that solar light reflected back off of objects. So anyway, that was slide 1. We have Alexa anyway. Another thing I did is, and I always do this because for me it really helps. Do we keep these doors closed, you think? I felt hot. Is it just because I'm excited about the sun? There we go. An outline. I'll put, so within my section on that desire to learn thing, that D2L thing, there'll always be the PowerPoints and there'll always be an outline. And I think that's so essential because you get lost in the detail and you go back and just see what were the big headlines. That's what I should know. So, oh, I'm doing that thing where I'm getting excited and I can't breathe. So we'll talk about that in respiratory physiology. So, something you could do, I suppose, is put your notes, like type your notes right in on that outline would be one way to do it because it would reduce the words you're having to type. And anyway, you just enjoy that. That's what you use and enjoy. Okay, so here's the outline. And it sits in the bony orbit. So you learned all of the bones of the skull, know them intimately at this time, right? Yeah. Didn't you just do that? Mm-hmm. Okay. And so do you remember looking into that orbit and just, it went down answer sphenoid? Right? So this zygomatic arch, you know that, this nasal bone, and you're just all seated in there so beautifully. Look, there's fat around that orb that we're going to, and in all the extraocular muscles, and that's really for another part of the course. And then where the artery and vein feeds in, inside that optic nerve. So that's kind of the big chunks. I like it. Okay. All right, so gross structure of the eye, the sclera, white of the eye, very opaque connective tissue. It serves to create sort of a shape, structure, rigidity, security for the eye. Do you remember in Meet the Parents when he goes up to buy that ticket and she's just typing away? This went to that right there. So the sclera is really only interrupted at a couple of places, and one is where that optic nerve exits, and the other is that clear cornea. It's just so beautifully structured. And you know the sclera is quite puncture resistant as well, although you'll see gross stuff later that proves otherwise. But you know, it's pretty hard to mess with the sclera. I'm going to look at my notes and see if there's anything else I want to say about it. Oh yeah, this is cool. It is continuous with the dura mater. So it's continuous around the eye, optic nerve, and back to the dura mater, covering of the brain. So you see this a lot. Tissue, we think of these as sort of separate entities because you sort of study them like that. But so many linings and membranes and such will be continuous from one spot to the other. So anyway, I think that's kind of interesting. The cornea, then, is a clear window in the sclera. It is an arrangement of collagen fibers so that they're running in one direction, so that you have clarity across that surface. And so that's quite brilliant. It's the only place that really light can access the entire surface, you know. So that's neat. And then there's an epithelial covering on it that pumps sodium out of that corneal surface, or corneal interior, because that draws water out, and that keeps those fibers real drawn tightly together. Otherwise, they could disrupt, expand, and it wouldn't be as clear. So that's really, it's cute. It's like it's all worked out to work perfectly, right? And then the conjunctiva, which is continuous with the rear of the eyelid, across the surface of the orb, and then continuous with the lower lid. And you know it's very thin, and if you've ever worn contacts, you pull it away from your eye, probably, try to pull that contact out. You don't like it when that happens. You like our conjunctiva to sit there. But it's a sliding membrane that will provide nourishment to the cornea. The cornea is not vascularized. There's no blood vessel there, or it wouldn't be really clear. And so it's one of those immune privileged things. That's why you can transplant a cornea without doing tissue typing, because the immune system can't really reach it. So anyway, it is the job of the conjunctiva to kind of help moisten and keep all of that working well. The lacrimation. I think this is so interesting, tearing. So look where the tear gland is. It's kind of superior and lateral and above the eyelid. And I'm not a super freak for knowing all of these cranial nerves, but you have to know all the cranial nerves. So I'm just saying, you know, there's sensory information, there's motor information. If you just keep that super clean, because it makes sense, you'll be able to get all this stuff to fall in line. So the lacrimation is sort of continuous to kind of keep everything moist and so forth across the surface of this eye. And also you can have increased lacrimation when there are dust particles. It's part of the allergy response because the nasal membranes will create more moisture. They will also block the secretion or drainage of that lacrimal stuff, and you'll have more tears. Or you can just have an emotional reaction and cry. Has that happened? Are some of you criers? I mean, you're either a crier or you're not a crier. And sometimes you just, it happens. You you know who you are. And there's that moment, because I don't cry in front of people really, unless it's something silly like fireworks, just, they just make me weep. I don't know why, but you know, I mean like, movies, whatever, I'm like a rock. But you know, there'll be these moments during your rotations, and I don't care how tough big old boy you are, you're going to be standing there and all you're going to be saying is, just don't cry. Just listen to him, yes, get to the bathroom, and later you can have a bath. So anyway, crying, sensory. That is sensory information in via the trigeminal nerve and out via the motor nerve. Kind of space on that gland, the little smooth muscles, and that can create lacrimation. Tears, and there's all kinds of studies about the content of tears. But we don't care about feelings. But I think this is cute. Tear film, it's not just lacrimal secretions, which is not just aqueous. There is this mucoid layer that lays against the eye, which helps spread the tear across the surface of that conjunctiva. Then there's an aqueous, very high water content layer. And then there's a lipid layer on the outside, and that helps the tear not spill over the edge of the lid. Unless, of course, the volume of secretions exceeds the ability of that lipid layer to retain that fluid level. Or you cry. That's cute, isn't it? And so anyway, there's the lacrimal gland, and there's this constant secretion and drainage via the lacrimal duct. The uvea is going, we're moving interiorly, and it's a three-part structure of the iris, ciliary body, and choroid. And so there'll be a whole group of sort of illnesses that are related. There's a uveitis and so forth, so you have to know it's really great where disease names follows the anatomical structure, and here you can see that. But the iris is the colored part of the eye. There are circular muscles, and there are longitudinal muscles. And so you can create constriction and dilation of the iris to open or close the opening in the iris, which is the pupil. You know that. This is interesting. The eye pigment really is just brown, but you have different amounts of brown, and so the reflection off of where that pigment is, if it's more forward or more posterior along that surface, how that color will actually look. And so I think that's just, you know, we think eye color is, anyway, it's one of the things we get emotional about, right, when we look at somebody, is their eyes, their dilation of their pupils, eye color, eye shape. It's one of these things that we sort of get to, and then we cry. Iris. The iris is controlled by the ciliary body. So the ciliary body will come up, and I'll show you a different picture of this. How big do these look to you? Oh, pretty big. So the ciliary body is coming around kind of along that edge of the iris, and will attach to the lens, and will attach to the iris. And so there's several things there. One of the things that the ciliary body does is, as part of its epithelial tissue or layer, will produce the aqueous humor, which fills this front chamber, or from the cornea to the lens, and we'll get to that. But it produces that, and that's continuously produced. And then there are muscles and ligaments that sort of hold and control all of this. So iris or aperture, and I have that spelled wrong on the slide that you have. I did a couple of corrections this morning, so anyway, I spelled aperture wrong. But you know, that opening and closing is a function of the ciliary body, and then the shape of the lens is a function of the ciliary body. Yep. Oh, iris means rainbow. Okay. And then the choroid is interior to the sclera and is this entire rear chamber. So when you look at this picture, you can see it's very dark looking for two reasons. One, it is vascular, so there's blood supply there. And it is full of pigment, especially melanin. And what that helps do is just absorb some of the light entering the eye, and that will help with bouncing of light off of structures, will decrease that and increase acuity because it will absorb light and kind of create a dark room in a sense for the photoreception to work better. Did you ever dissect a cow eye? You just dissected some human eyes, didn't you? Right, wow. No, you didn't get into that eye. Have you ever done a cow eye back in? I remember the choroid. It was just this kind of blue, sort of pearl eyes. It had a little iridescent kind of look to it in the cow eye. Yeah, it was always sort of a favorite part of the eye for me. It's weird. Okay, lens. This is really a big deal. As we age, the lens gets replaced. You know, that cataract will cloud the lens, and they get replaced, and this is why cataract surgeons are very wealthy. Of all the surgeons, they just make all the money. They really do. And it's really cool because that lens is enclosed in a capsule. The lens isn't actually attached to anything. The capsule surrounds the lens, and its suspensory ligaments attached to ciliary body muscles that help move it. So it's something you can make a little slit there, pull it out, stick a new one in, and it's just an amazing thing. And then people can see. So when you have that as an ability in your insurance plan or the country you live in, you know, it just extends the quality of your life so much. It is biconvex. That was one of those words that always bothered me. Convex and concave. I don't know if that's one of those that gets you. But concave is like a red blood cell. And so if you only remember one, you know the other one's opposite. Do you do this? This is a good thing to do as a tool. When there's one thing and it's in opposition to something else, don't remember something else. Remember one, know one, 100% for sure. And then you can always deduce the other one. Or you'll be going, wait, okay, do I get it backwards this way or do I get it backwards this way? So it must be this. I think it's that. Well, I'm guessing myself. I'm probably wrong. Oh, dear God, I'm going to die penniless. I mean, I hear people think like that. Probably you don't. I mean, I don't. Context. And so it can flatten and round a little bit, and that allows for refraction. We'll talk about that a bit more. Nutrients, there is no blood supply to it. The protein that is in there is called crystalline protein. It's a certain kind of protein that really will appear clear. This is important because we have to let this light in. So we really like the fact that this cornea and this lens allow light in because they have just a really unique and special sort of arrangement of protein. Because when you don't cross-mesh stuff, then you can let light in. And so that happens there. The lens with age, those crystalline proteins, the protein fibers, will become more dense over time. The lens, those are constantly being made and renewed. And so over time that kind of lags up, it doesn't work as well, and that's why there are cataracts problems because the lens clouds over time with age. And then some other things will do this, exposure to UV light, wear sunglasses, you're cuter anyway, and then also stuff like steroid and some drugs will have that eventual clouding of that lens. So anyway, let's see. Nutrients from the aqueous humor, we'll get to that. So again, suspensory ligaments attached to ciliary muscles, and this pulling and rearranging can happen on the shape of the lens to allow for accommodation, and we'll talk a bit more about that. Refraction is another job of the lens. Refraction is just meaning that light is traveling along its wavelength to a particular medium, and when you hit another medium, whatever that is, it will change the direction of that wave. And so what we want to do is, because our eyes aren't this big, and we have to see a world that's this big and an opening that's only this big, we want to make sure we can get all the light in there that we can. And so the cornea helps bend and refract light waves, and so does the lens. And then we'll do more of that as well. Interior chamber is that spot. This is kind of a weird picture. You see the cornea up there, and you see the lens. So the anterior, you only see half of it. So the anterior chamber is between the lens and the cornea. And here you really get a good glimpse of this ciliary body with its ciliary epithelium and the iris coming out as sort of a part of that extension of that ciliary body in a sense. And you see these suspensory ligaments attached to that lens capsule and muscles there. So that ciliary body and anterior chamber between the lens and the cornea, and it's fluid. It's a fluid-containing chamber, and that fluid comes from the ciliary body. It's made constitutively throughout life. There's a continuous production and drainage, and so that has to be kept at a certain rate. When there is any obstruction to that damage or to that drainage, and production doesn't cease, then you can have an increase in the ocular pressure, and that's one of those things that they test for, and you're supposed to have that tested routinely over the age of 40, is it? Or is it 50? I think it's 40, because increased pressure is glaucoma, and so that is painless. There are some painful kinds of glaucoma. That's a different kind of category as well, but a glaucoma that is painless and will eventually cause blindness. And so I think it's really going to be interesting and something to look for, because so many people have LASIK surgery, have the correction for their eyes, and aren't getting the routine eye examinations. And so it'll kind of be interesting to see if we have an increased incidence of loss of vision due to glaucoma that's been undiagnosed, because everybody got that test at a certain point yearly when you go get your glasses checked. So anyway, that's just, it's another thing I said more about. Blind people. Anterior chamber. And you know, as PAs, we're really just not, there's so much about the eyes we're not supposed to do, so that'll be interesting. I mean, in a bad way. Interesting. Let's see. So cornea and lens are both things that can be transplanted. Anterior chamber. Oh, that was random, wasn't it? Okay. Anterior chamber. Posterior chamber. So the eye is this sphere with all of these layers, and then the chambers. Anterior, aqueous humor, constantly changing. Vitreous chamber is really this big part that fills from the back of the lens all the way up against the rear, the roof, and the floor of the orb. It has, it's a vitreous humor. It's produced by the ciliary body, but embryonically. And so it's stagnant, which sounds like a bad word in a way, but it just means that you're born with it and you die with it, unchanged for the most part, unless little bits of something are floating in there, and you just get to see those every time they flip by. So that's interesting. That doesn't change. It's just one of those things you're born with. It is thicker, and so here is it looks like a human eye, based on size. And you see the sclera and choroid body have been opened there, but that vitreous humor has some hang to it, you know? It's got some shape to it. And so this is kind of another one of the many reasons that it's, you know, I don't know if you thought about stuff like this when you were a kid, did you ever think if you got poked in the eye, your eye would just drain out and you'd have like this big wet drainage or anything? It's just, I mean, I didn't think like that either. There were other kids. There was some fears and some issues that didn't enter my family. We're talking about ocular health at the dinner table. But anyway, but it's really not that way. So even when you do see ruptured orbs, and if you get to see one in an ER, I never got to, but just pictures of them, there is the orb holds together very well because of the sclera and the vitreous humor having some oomph to them, which is nice. Another thing it does is, you know, the retina isn't attached to anything. It's just sort of blown up against the wall because this vitreous humor is there pushing against it. So if there is some sort of leak in the vitreous humor, and a bit of it can happen, a bit of it gets between the retina and the choroid layer, then that retina can detach or move. And so that can be bad. And, you know, a lot of eye rest can kind of help heal that, or you can have loss of vision or part of vision with that. And they're doing some neat things where they inject a bubble in there to kind of create that pressure and push that retina back up against the wall until that gas gets eventually reabsorbed and the whole thing sort of situates itself. So that's kind of a neat thing that ophthalmologists do. Inject bubbles. I've known a few people that had bubbles injected. I don't mean to brag. I wonder what this big middle button does. Can you see that?
It's small. The TV's reflect it.
See, you can't see what I see. But your eyes don't see what my eyes see. See, they're different. Mine and yours. Hey, that's okay. Does that work?
I don't think it'll go through the.
That's what he's
People have been looking for a laser pointer and they couldn't find it.
But I can't work. So you can't see it there? So you don't have to hold it.
No.
That's not helpful at all. Okay. Retina. That's weird, isn't it, that you can't. Okay. Retina. No, it's the business end of the orb. I like saying orb. It's hard to work into ordinary conversation, however. You're going to do it, and no one will invite you back over. At least that's been my experience. Okay, looky there, sclera. Choroid. See how that's kind of bent forward? I'm just going to point out it for myself. Y'all can't see it. It's going to be awesome. I just enjoy it. Is it just on the wall? See, here's your choroid. Okay, so that vascular layer is peeled forward, and then you see the retina. And the retina isn't just the back, but it really does extend all the way forward. And it's a large tunic or layer. It's where the photoreceptor cells are. It's the neural layer. There are a couple of flavors of photoreceptor cells. Those are modified neurons. They're rods and they're cones. And we'll talk more about them. The rods are densest to the periphery, and they are mixed in less and less as you go toward the rear compartment of the rear section of the retina. And the cones become denser and denser until you get to that fovea. This isn't named here, but the fovea is the center of the macula. And that word is there, macula. And that will be high concentration of cones, with the highest concentration of cones and zero rods in the center of that macula, or the fovea. So the fovea is inside the macula. It's also called the fovea centralis. It's the most acute, bright color vision site on the retina. And it's really where we spend our time trying to focus images. We're going to have the best delineation of shape and acuity of detail right there. And acutely, the neurons that are taking information from the retina are really kind of pushed aside there so that you can actually get the most light to that spot, and that's why it's associated with the greatest visual acuity, which is just insanely cute to me, because the neurons make the whole thing work, but they get out of the way to make it work better, in a sense right there. Not like they're moving, but it's less dense there. The optic disc is the portion of the retina where the optic nerve is exiting. Look at the optic nerve. What cranial nerve is that? You don't do those yet, but it is to your. Have you done those yet? Yeah. Oh. That was a good time. That's another one of those things, isn't it? Okay. On top of Old Smoky or something, that thing where you remember all of them. You see the central artery and vein for each eye is really right there in the center of the optic nerve. And then those blood vessels will spread out, and you can see them on a fundoscopic exam. Also, there's this retinal pigment epithelium, which is a layer of the retina. So while most of the retina is neuron, there is this epithelial layer, and let's look at that, shall we? Let's do. Okay, this is so weird. I feel like just looking at the screen rather than you guys. Okay. So this is the thickness of the whole retina, the layers of the retina. So look all the way to the back or to the right side there, and you see pigment epithelium. You can also see it in the first chunk too. This cuboidal epithelium that is the posterior most layer of the retina. It will, it's cute because it also has pigment granules in there, melanin, like the choroid layer does. So it will just be sort of light absorptive, and light kind of goes there and dies, and that helps visual acuity. Also, it will have, those cells have the ability to phagocytize and hold vitamin A. So they're getting it from the nutrient supply. You eat, absorb that vitamins. A is a fast-moving vitamin, and it goes into the whole liver, and it goes into the liver proteins and so forth, and it gets served to the eye. And because it's so important for it's the visual pigment, you have to have plenty of it so that epithelium will actually store it. And that's cute. And so you can replace especially the visual pigment in rods throughout life. So even though it's a neuron and those things can't be fixed, you do replace parts of it. So a constant supply of vitamin A is essential for vision. And so one of the number one worldwide reasons for blindness is lack of vitamin A deficiency. Vitamin A is also really important in epithelial tissue, mucous membranes, and maintaining those membranes. So really going to the moisture and the membranes that cover the eyes as well is important. Vitamin A is an important nutrient for the health of that tissue.
What kind of stuff would the cuboidal epithelial cells have to phagocytize if the inside is kind of stagnant?
Okay, they're part of the retina. And so are you thinking of vitreous humor?
Yeah.
Being stagnant? Yes, it is stagnant. It just kind of sits there and presses against the retina. So we're in this retinal layer. So the retina itself has its own blood supply coming in kind of on that front part. So these are retinal layers. And the back of it will have this epithelial layer that will help service the rods and cones, or the photoreceptor cells, and then will hold pockets of vitamin A there as well. Does that do it for you?
I guess I was wondering, because you mentioned they can phagocytize things, and I was wondering what you would have to phagocytize in the eye.
Gotcha. It is, what they are phagocytizing specifically is vitamin A from the blood supply and holding a reservoir of that to maintain the rods, especially, and cones as well too. So to maintain the photoreception molecule, part of that, the whole series, you have this ability to store right here. You don't have to wait for blood to process through. You're holding on to some. So I think that's kind of cute. And it is a fat-soluble thing, so it's kind of easy for it to keep and not to destroy. It shouldn't, be degraded within that tissue as well. Is that clear?
With the vitamin A deficiency, is it restorable or does it lead to permanent blindness?
It's restorable to a point. And I can't tell you more than that, honestly, because I don't recall. And I'm really trying to decide if that's age-related or it is disease-related, and I can't really remember. But if you can restore generally, if you can restore any vitamin deficiency early enough, you can correct some of the disease. And with vitamin A blindness, you can correct early on, like you can correct night blindness and stuff like that, but if you've actually had destruction of visual photoreceptor cells, you can't fix that because those don't regrow. So that's a half-ass answer, how's that? But you know, isn't that something though, when you think about something as simple as, I mean, don't we all just pee very expensive urine because we have really good diets? I mean, you know, we have such good diets. And then so, really, I mean, even, look, I mean, everything's fortified. If you're only eating, you know, what you can only eat. But I just, everything's fortified. There's so many vitamins everywhere. And to think that the majority of the world, the cause of blindness is vitamin A deficiency. So it's a big deal, and those kind of deficiencies are huge in youth as well, because then you parlay that through life, loss of all through life. So, and sanitation's huge. Another cause of blindness really is parasites. Anyway, it's so simple. Our lives are so simple that we've ruined them by complicating them. And I like to think we do it better than anyone. Okay. Vitamin A is stored in that pigment epithelium. So let's go out at the other layers of the retina. So you have this pigment epithelium here, and there is the blood supply, the vascular blood supply here, which would be anterior. So we're looking back toward the back of the eye. And this is always cute and weird to me. If you've seen this before, you maybe thought so too. But the photoreceptors are the farthest away from the light. And so you see in this, the simple view over here, where you see the rods lined up, they synapse with a neural cell called a bipolar cell, which synapse with ganglion cells. And the ganglion cell, like you might think, a ganglion, a collection of tissue outside the central nervous system, or a collection of nervous tissue, will send axons to basically the brain for sensory information. And so the light is coming in, look over here, see the light coming in actually through this sort of transparent layer, transparent-ish layer of neurons to the photoreceptor cell. So you'd think that photoreceptor cell would be up forward, but it's kind of faced back. So what it's really doing is getting light that bounces back off of that choroid and pigment layer. So you really want to absorb a lot of light before the photoreceptor cells begin to sense it. That's the only thing that we're able to see because of is the fact that these photoreceptor cells see a photon of light and undergo a conformational change, and then it gets complicated. So the photoreceptor cells, rods and cones, you see them both in this side. Rods and cones. So rods have a more rod-like shape, more cylinder shape, and a cone has a little bit more of the apical end is a little more cone shape. So that's where they get their fancy names. And then they're synapsing with a layer of cells called the bipolar cells and the ganglion cells. And there are more photoreceptor cells than there are bipolar cells. There are more bipolar cells than there are ganglion cells, which means that a lot of photoreceptor cells are sending information to a single ganglion. And so that's how we're able to have so much detailed information at all, really. That kind of coalesces. So we say that's convergence in the retina. That a lot of photoreceptor information, it converges into a fewer number of ganglion cells. Those ganglion cells have an axon that makes a right turn, or I should say a 90-degree turn. And then that will exit to the optic nerve for processing in the visual cortex. That's how you say visual cortex. Visual cortex. And that whole process is still not well understood. It's just so crazy. The more you learn about this, the crazier it all seems. Okay. We'll talk way more about that function. That's how it's set up. Retina. Oh. The ophthalmoscopic exam. Now, do you guys buy your ophthalmoscopes now separately? Did I ask this before of you guys, or somebody used to buy our own? So all of our friends and family super hated us because you're just trying to look at people's crap, and they don't like it, but anyway. The fundus is the part of the retina that you can see in an ophthalmoscopic exam. And a lot of diagnosis can be made from this if you get proficient at it. And if you don't, then you refer them to somebody, and you're going to anyway. But there really is, you can visualize the vessels, and so you can visualize things that have gone wrong with those vessels. And a lot of things, because there's a heavy vascularization in diabetes, for example, and then so for some hypertensive illnesses, you can see kind of a lack of vascular, so forth, vascular networking. The fovea and the macula. So macula big, fovea little, you can kind of see those there. And if you have an eye exam, really they're doing a digital image and then looking at the fundus digitally now on the screen. And so it's really better because you're not just hoping you can see what you can see, but you know they dilate the pupils so that they can see in there well. And that's something that PAs don't do, is dilate pupils. That's a part of our law. But you will be doing some ophthalmoscopic exams. It's just one of the things I wasn't really good at. You'll have something you're good at. I can sew like nobody's business, but you know, that wasn't mine. And you know, you'll all have your thing. So anyway, the fundus is what you can see. It's the only part of the central nervous system that you can visualize non-invasively. So the retina is an extension of the brain, really. And so that's cute. That might make you enjoy your ophthalmoscopes more. The visual pathway, how then is the information received and sent? I wonder what slide number I'm on. Let me do a little
15.
What is it?
15.
Oh. You got your photoreceptors. This is redundant. Several of these slides are quite redundant here. You got your photoreceptors because there's some picture that will make sense to everybody. All right, so that's why I try to put a lot of them up here. So rods and cones, and you see their business end is really kind of buried in that epithelial side. It's kind of there, and that's where the photoreception occurs. And then they will get excited and stimulate the bipolar cells, which will get excited and stimulate the ganglion cells. And that is the pathway from photoreceptor cell to bipolar cell to ganglion cell. And these will be electrical stimulus, electrical signals, because it's nervous tissue, and that's kind of what nervous tissue does. Nervous tissue uses electrical signal or neurotransmitter, and you know, with neurons you get that. None of these generate action potentials. Oh, do you hate that word? Do you love the word? Do you hate the word? Action potential, graded potential, all that stuff. These cells depolarize, but only the ganglion cells send an action potential. The difference between a local graded potential and an action potential is really just distance. And so because the neural cells are located very closely together, these are just going to be local graded polarities that send information. But you're going to send information to the visual cortex via the ganglion cell. Now there's another couple of things that happen, and you know this happens in nervous tissue too. Some lateralization of information. So photoreceptor cells are just in their own line, but when you get to the bipolar cell layer of the retina, there are some horizontal cells that will synapse one bipolar cell to another bipolar cell. Those are little neurons called horizontal cells. They're not shown in this particular picture, but I have another one that does. The ganglion cells have these sort of horizontal kind of connective interneurons called amacrine cells. And I'm not going to talk about the function of horizontal amacrine cells because it really just goes beyond what I feel like is important to know. Except that it's just part of that processing of information, and it's part of that convergence where you have billions of photoreceptor cells and millions of ganglion cells. So information has to be coalesced. So there's a lot of excitation, there's a lot of inhibition. And this is really all you've got with nervous tissue, is excitation or inhibition. And how that all works out to a cerebral consciousness and vision, I don't know. It's just off or on. But this integration of information happens and is enhanced via these interneurons, the horizontal cells and the amacrine cells in their respective bipolar and ganglion layers. Ganglion cells, which will be the thing that actually sends the signal to the brain, also has a photosensitive. There are photosensitive ganglion cells there. They just sense light and dark. So there really are three kinds of photoreceptors: rods, cones, and ganglion photosensitive cells. And that goes to light-dark sensation. And that has its own thing, it kind of is handled elsewhere, it's part of circadian rhythm. And so that is going to be sensed where the light comes in, that makes sense. But it's not part of the visual field or visual information. And then the optic nerve is the collection of ganglion cell axons. And remember, we're bilateral. How many times did we have to remember that? We're bilateral. And Dr. McNeil says that a bit, doesn't he? That you do coalesce information from each orb. There is decusation of information, and then you go to different hemispheres. But we have two eyes, and so we do take and assess both of them together as well. So exciting. Oh, I guess. Okay, here we go. Convergence. 150 million receptors, 150 million rods and cones, but only 1 million nerve fibers, meaning ganglion cells. So that's a lot of processing before the signal is sent with inhibition and excitation. And that goes to how we're able to create this mosaic that makes any sense of the visual cortex. So there's just another picture. What was I going to say? Oh, I think it was this particular picture does show these interneurons in the bipolar and ganglion layers. Rods and cones, and then the bipolar layer with its horizontal cells, so you see them there in that picture, them there, and then the amacrine cells for the ganglion cells. So there's a lot of interpretation. Every layer is processing what the layer before it is sending until you get to the ganglion cell. Okay. We'll get to how that all works, but first let's discuss light, shall we? Refraction. So I mentioned this. The inotropic eye is the normal eye. No kind of enjoyers. And so we're really looking at a focal point in the normal eye. And we can correct these things, you know. So there's the light source. It travels in an unchanged path until it reaches a new medium, and light rays are bent when that medium has a convex shape. The rays tend to bend inward, and this is how we can collect a lot of light by using this convex shape of our cornea. I don't know why I don't want to use that word today, cornea, and then also the lens. So we're reflecting as much light as we can, or we would be missing tons of light rays. So we're sort of focusing in. That's what your light microscope did. You had the light coming in, and there was a condenser that just bent the light rays up into the little tiny end of that microscope objective. And so that's really what we're doing. We're taking these light rays and bending them in, trying to get them to converge inward. So refraction is via the cornea and the lens. Accommodation is to further bend them, to further create a focal point. And that is the ability of the flexible lens, because it is attached to, there is some muscle control there. And the lens can kind of flatten and it can kind of round. And so that creates accommodation. the cornea can't accommodate because it has no ability to squeeze, but the lens does. Oh, here, this is different. I think I corrected this. Look at what you have. On your slide that you're seeing there, under accommodation is via the lens, it should say the muscles constrict and the lens rounds, but I believe your slide might say the opposite.
Mm-hmm.
So just make a note, or, I don't know, can you change slides on there?
Just fix it.
Just cut and paste and it was all crazy. But that makes more sense, that when muscles constrict, you can kind of pull on the thing. So we have the ciliary body muscles, and there are these suspensory ligaments attaching to the capsule around the lens. And they can constrict and pull on that lens. When they constrict, the muscles will get long and pull on that lens. And it relaxes, it flattens. So our set point for vision is for distance vision. And a normal emmetropic eye can see clearly at 20 feet. So that's where that 20 thing comes from, 20/20 thing. You're at 20 feet, a normal, relaxed lens should focus at the back of your retina. That's the focal point. You've converged and accommodated. And when you're seeing distance vision, the lens is at its most thin, the muscles are relaxed, and it's flat, so you don't have to do a lot of accommodation. There's just some refraction to stuff it close. You can see it pretty well and make a focal point. Then things change when the eyeball shape isn't quite right, or that lens doesn't accommodate well. And the focal point shifts based on where stuff is in your eye. Okay, again, that is another one of those where relaxed ciliary muscles should mean a flattened lens. So that's something you need to correct on your slide here. Okay, first picture, viewing a distant object. Those light rays, that reflected light, traveling pretty parallel and entering the eye well without a lot of need for accommodation. There's just the refraction that's default refraction at cornea and lens, and the focal point's where it should be. Now, if you're looking at something near, you have to do a lot of accommodation and bend those light rays in. And so that you make this rounder lens to do that and get the focal point where it's supposed to be. So if it's flat and you're not refracting much, we say that's weak refraction. For reading close, for getting close, you have to have strong refraction. Until you've done it so much that you just can't refract anymore. You've refracted as all you can, and then you can get glasses, typically, and this is pretty easy to fix. So here's these things. This is one of those. Just remember one. What's all this paper here?
For our tests, paper.
Didn't use much, did you? That worked out. Okay. Normal eye, an emmetropic eye. Look, the light rays come in, they refract, they accommodate, and then down to a focal point right on the back of your retina, and bam, your vision is so good. But if you're myopic, that means you're nearsighted. You can see near, myopic vision. And that the eye shape is sort of long, and the focal point ends up in front of the retina. And so we want to move it back, and we use a concave or a biconcave lens that will just help move that focal point back to the back of the retina. And I'll show you that picture in a minute. Nearsightedness, myopia. I don't know what else I could possibly say about that. Myopic vision, your eyeball is long, but your vision is short. Oh, I like it. Hyperopia, farsightedness, short shape. You can see far, but you're bald. your ball is short. Short orb. Such a short orb. Do not, stupid. But the point, it's just so random, you know, because really, this is boring, right? It's a little boring. Let's make it exciting. Oh, do you need pee?
Yeah.
Go pee. Come back. I'll start at 2. You have 5 full minutes. Enjoy.